So I'm pleased to introduce our next speaker, Professor Dr. Martin Korte. He is the Director of Zoological Institute at Technische Universität Braunschweig. So today he will share the insights on communication between neurons, synaptic activity as a molecular correlate of learning as memory.
Thank you. Thanks for having me here as a neurobiologist talking to you. I'm not sure if I got the right level to talk to you. The main topic of my research group is learning memory and also forgetting, and also in order to store information, neurons need lots of signaling pathways, and I think that what brings me in here. And for learning and memory from a biological perspective, it's of tremendous importance because of whatever our brain does, whatever we experience in the now, in the past, but also when we plan for the future, we need a memory. But that also means that the nervous system must process a continuous flow of sensory information, which, except when we sleep, basically never stop. But it must be at the same time be able to store memories, sometimes for a lifetime, and adapt the animals and, of course, also our human behavior accordingly. And this is what this is all about. That's why the signaling pathways are so complex, and also they have to be very secure in terms that they have to keep the balance between change and stability. And as I said, this is what my lab is working on, which means how can signaling pathways in and between neurons secure that information can be stored. And there was a crazy idea many years ago, actually 65 years ago, in saying that, well, the easiest way would be to store information how the immune system does in terms of single proteins, because an antibody produced by B memory cells in the immune system recognizes a bacteria or a virus or any kind of parasite that has invaded our body before, which means the shape of the antibody has a memory of the receptor, of a certain receptor, or of any component of the surface of a virus or of a bacteria, like for SARS-CoV-2, it's this famous spike protein. And we do have B cells that produce antibody which precisely fit to that, due to some immense genetic engineering that happens after we survive the first endeavor with encounter with the virus. Neuroscientists at that time, in the end of the 1960s, had basically no idea, not even conceptually, how something like an idea, something we read, something we learn from a location can be stored in the matter of the mind, which means in the brain. So after it was discovered how the immune system died, neuroscientists at that time thought, well, cool idea. Probably the brain also stores information in single proteins. And they have done a crazy experiment, a very nasty one. They have trained rats to avoid the dark. So if you have an open cage, like the size of the desk here, half is dark, half is light, the rat will immediately go to the dark, just to avoid predators from above. It's a natural instinct. But if they get an electric shock in the dark, after a certain amount of times and rounds, they learn to avoid the dark and to stay into the light. After they've learned that for sure it didn't do them any good, because they were immediately killed and the brain was fed to other rats. And then the rats were trained naive to the situation, and they learned actually much faster, which meant the scientists for a second thought they have proved that it must be proteins where memories are stored, and they can actually feed it to other rats. The molecule was called scotophobine, a protein that helps you to avoid the dark. At that time, neuroscience was not a very successful endeavor in terms of biology, so it rarely made it to the headlines of major newspapers. But at that time, when the paper came out, the New York Times had a headline saying, well, if that's true, what we can all basically do is we don't have to learn anymore. You don't have to travel here. You can basically, the lecturer would send you a pill with this lecture and you can eat it. it was a cool idea for a startup. In the end, it turned out what the scientists have fed to other rats was basically a stress hormone, and the stress were more alert. Proteins, if they are not prions, do not survive our acid situation in the stomach. So it turned out to be, in the end, one of these experiments that have been proven wrong. But also, people actually didn't follow up on it because the perspective changed in a way that people thought, well, we can recognize 80,000 to 100,000 words. We use 20,000 words in our active word memory. We can memorize 10,000 of pages of faces, not pages, and so on and so on and so on. And we have 20,000 or 22,000 genes. So simply, proteins are made out of genes. It doesn't work that way. So people then got the assumption and were following, and we are still following that path. Memory storage is a network property. Information are stored between neurons. So here you have a typical pyramidal cell of the cerebral cortex. We have around 14 billion of that. If you think that's a lot, think about the cerebellum. There are 40,000. There are 40 billion neurons, so a lot more. On the surface, the excitatory neurons at least do have these protrusions. These are the postsynaptic side of the receiving end of dendrites in neurons. You can stain them, you can analyze them, and what my talk will be about is about here what happens at the synaptic side, because when we learn something, synapses change their strength of submission, they can even change their structure, which means when we would like to understand how the brain stores information, how neurons can do that. These are the dendrites I've shown before. Here's the soma, where the nucleus is, the DNA is. Here you have the long axon, and then here you have the presynaptic side of synapses. We actually do have two flavors of synapses. We have electrical synapses, which are tremendously fast because they are connected to each other, and if there's some flow of ion, that's the transport of electric charge in a neuron, then they can immediately go through channels to the next neuron, which is fast, but it's also a very stupid way of transmitting information because you cannot change it. So there's a second way, much slower, still one of the fastest biological processes described, but I will go into it. That's the chemical transmission. And this one is modifiable because you can change it on the sending side and also on the receiving side. So my talk will now focus on the chemical synapse. I will ignore the electrical the fast electrical synapse, but also that has its role. So the network of the brain is not so complex because our brain has, in humans, 83 billion neurons. It's so complex because it has 15 trillion synapses, and you can change the weight, how much information is transmitted from the receiver, from the sending to the receiving side, and also the structure of the brain can change. Here you see an EM picture of such a synapse, color-coded. It's a very energy-demanding process, so you have lots of mitochondria, you have vesicles where neurotransmitter molecules are stored. Here you have the synaptic cleft. It's tremendously small, just 20 nanometers. It's smaller than the vesicles, actually. And as I've said, for biological process, it's fast. The brain synapses send information at around 120 meters per second, but in terms of the speed of light and in comparison to any current computers, it's extremely slow. But for a biological process, it's actually fast. And if you think about it, when we react to a stimulus or avoid, or if we are predators and would like to hunt something, we also have to react in a certain fast way, let's put it that way, for a biological system. But the processing speed of the brain depends on how fast synapses are. The movement of ions, action potentials, and so on are in comparison fast, but in the end, it depends on how fast molecules get from one side to another. And actually, when neuroscience started on the cellular level, which was at the end of the 19th century, beginning of the 20th century, there was a famous neuroscientist, Ramon y Cajal, who actually proposed that neurons are entities which are connected via synapses. He had no EM at that time. That was invented a lot later. He just deduced it from a staining method, which is now still used. It's still used today in my lab. It's called the Golgi method. It was invented by the Italian Emilio Golgi. And Mr. Golgi, with his method, actually thought, no, the whole brain is a syncytium. There are no synapses. And the funny thing is, both got a Nobel Prize at the same year, 1906, and they were almost getting into a fist fight when they got their Nobel Prizes because Cajal, with the method of Golgi, showed that Golgi was wrong. And Golgi, of course, never admitting being wrong since he invented this method. But in the end, Golgi was right. We have all these synapses. Why is the process so fast and why is it so secure? Couple of things you can see in this picture. This is now here on the upper side. That's a synaptic vesicle close to the membrane. I go into it. And here you have the membrane. And the presynaptic membrane is in particular prepared in order to get the vesicles to fuse with the membrane. So two lipid membranes have to fuse with each other, as you can see here. But they have to wait for a signal to come in order to do that. And they need a calcium signal. So Nobel Prize was given for that to Tom Süthoff, who actually was part in his life in Göttingen, then went to Texas, is now in California, and was actually at a certain stage supposed to come back as a Max Planck director to Göttingen. The animal house was already built. It was the largest one the Max Planck Society ever built. And the day he was supposed to show up as a Max Planck director, he actually didn't. So that's why you got this Nobel Prize when he was still in Texas. And what he could show is that the calcium entry here is the trigger when or actually a couple of proteins were then changing their 3D structure and were able to interact. So the binding site of these molecules were exposed, of the T snares, V snares, of synaptotacmin, and what the name of the molecules are. And then with the machinery that actually drags the lipid membrane of the vesicle to the lipid membrane of the presynaptic site, the vesicle molecules are released into the synaptic cleft. And since the vesicles are very close to the presynaptic membrane, it's a very, very, very fast process. Here you see it again. We have a couple of molecules which are already in the vesicle with the neurotransmitter molecule. We do have vesicles which are only present in the presynaptic membrane. They are all close to the calcium channel, and then if calcium enters, they can intervene, interlock, and then in a kiss and run mechanism, the interior of such a vesicle, the neurotransmitters, glutamate, could be acetylcholine, could be GABA, small molecules which have been transported into the vesicle before. And that's actually a very energy demanding process because you have to put in such a small vesicle 400 molecules against the concentration gradient of the in and outside. So you need lots of ATP in order to get that done. And here are only a subset of molecules that you all need in order to have all the vesicles lined up. They're like little soldiers in one of these Woody Allen movies, which are now ready to be released and now wait for the signal to come. And then the process can then start. So 10 to 20 vesicles will be released at the same time. For example, for glutamate, it has been shown that with one event, around 8,000 glutamate molecules are released into the synaptic cleft. But then when the action potential is gone and the membrane is not depolarized anymore, the depolarization-dependent calcium channels close again, and then the next signal can come in, and the synapse can do this 100 times in a second. And then also in the meantime, the glutamate has to be removed from the cleft. And part of it all depends on this vesicle. And you see here the structure 50 nanometers large, which is now analyzed for years now by Reinhard Jahn, who actually is a Max Planck director in Göttingen for many, many decades now, for many moons. He's actually retired, still working on an ESC grant and going on. And his group has put all data together and has done let's say, a landmark of such a vesicle. Here you see the important ATPase that's necessary in order to pump all the glutamate or any other kind of transmitter into the vesicles. And there are just 2 ATPases on a vesicle, on each vesicle, and there are thousands. If they are missing, it's all a waste. If there's 1, it's not enough. If there are 4, it's a waste of energy. So there are always 2. And you also see that the lipid membrane is completely covered with these signaling molecules. Here, if you look inside the vesicles, you're actually wondering where the neurotransmitters are actually, because it's all stuffed and it's all full, in order to have a regulated process which is very error prone and is very reliable in order to make sure that that after an action potentials enters the presynaptic terminal, that it's ensured that this vesicle is released. Here you see just one of the important molecules, the synaptobravin, which is important for the combination of the vesicle membrane with the outer membrane of the cell. But then that's only half of the story. Then the neurotransmitter is released into the synaptic cleft, but then it needs to do something. And this synapse is designed in a way that on the other side, 20 nanometers away, you have lots of neurotransmitter receptors. They all come in different flavors. The fastest ones are the ones that are directly combined with the channel that in this case opens for sodium, and sodium then leads to an extra leads to an event potential that to an extracellular potential that depolarizes the cell. You can also have inhibitory receptors that are coupled to a channel that leads chloride into the cell, and then you have a negative effect. Which means to look at the whole picture, I have an axon, there's some nerve pulse and action potential coming in, calcium channels open, neurotransmitters are released, and they can bind to the postsynaptic membrane to ligand-gated channels which open, and then the signal can be transmitted via the whole dendrites to the soma, and if the signal is strong enough, it will elicit an action potential in the next neuron. Here you see also only part of the picture, but you see that in order to make sure that this synapse stays together, you have molecules that are bridging the gap. For example, ephrine receptors, you have neuroligin, you have N-cateins, all kinds of molecules that make sure a certain distance between the synapses is ensured. And on the postsynaptic side, you need something which keeps the membrane in their place, because this lipid bilayer is kind of a swimming area, so they would all move out of the synaptic cleft if there would not be molecules that would hold them in place. As I've said, these synapses are instrumental for learning and memory processes. For example, in this red structure here in the hippocampus, which is the brain structure in the mammalian brain, so also in the human brain, that's important for all fact memory, for autobiographical memory, and it makes sure that the memory is in the end stored in the cerebral cortex, but also partly for a certain time period in the hippocampus. So this is a good place to start. And the neuropsychologist Donald O'Hebb thought about a rule how information can be stored. And what he actually said is, to put it into a nutshell, if 2 neurons are connected to each other and they fire in a time window which is very close to each other, optimally at the same time, they will actually show some metabolic changes. I would now call it functional changes, or some structural changes. It took until 1973 when Timothy Bliss and Mr. Anderson were able to show, in that case, in rabbits of all cases, that such an event can actually happen under circumstances that Hebb described. So they termed it long-term potentiation, and it was measured in the hippocampus. You see here the baseline recording, which at the same strengths at a certain time interval, synapses going into the hippocampus were stimulated, and then at the same time, pre and post synaptic neurons were stimulated, and in the end, with the same stimulus strengths that have been used here, the synapses have been strengthened. They can also be weakened, so the same synapses, you can basically pull the dial in two directions. You can pull it up, you can pull it down, which is then termed long-term depression, and both can happen, as I said, at the same synapses. This rule is also to make it easier to memorize. Neurons that fire together, wire together, neurons out of sync fail to link, because this mechanism that Mr. Hebb described is not only important for learning and memory, it's also important for the development of the brain. And the next question, of course, would be, well, you have a stimulus at the same time, and then the synapse becomes stronger. It's like the scientist being at the board, writing down a complicated mathematical formula, and in the middle he writes just a miracle occurs. So what's actually happening? How is that achieved? And we do have a coincidence detector. It's a glutamate receptor. It's a subtype of the glutamate receptor. It's called the NMDA receptor after one of its agonists. That receptor is usually closed. The one that always opens if glutamate is released into the cleft is the AMPA receptor. That's another glutamate receptor. And the NMDA receptor is closed because magnesium blocks the channel for sodium. So this channel only opens when magnesium is kicked out of the channel, and it's only kicked out of the channel if the membrane is depolarized. Which means with this elegant mechanism, we have the release. Basically, if you have a Pavlovian dog, you have like an entry of, let's say, a noise from a bell, and at the same time, you always, or closely after, you get some food. That means if you have two different entries to this cell here, it means the cell is depolarized when this one here becomes active, which means at that time period, the NMDA receptor can open, and then with this coincidence detector, calcium can enter the cell. Calcium is in biology the most important second messenger, and that then can lead to processes that strengthen the synapse. So here you see it again. Here you see what I described before, the baseline synaptic transmission without any activity of the postsynaptic neuron. So there's no signaling here. So if this signal integration is active as well, when glutamate is released, glutamate binds to the AMPA and to the NMDA receptor, magnesium is removed because that's indicated in red here. The cell is depolarized, calcium can enter the cell. Signaling pathways go on. The most important activator is the CAM kinase 2. That's the molecule which the highest expression level on the postsynaptic side. And this now ensures that new receptors are inserted into the membrane of the types that always open if glutamate is released. So if we are now back in this situation, we have more AMPA receptors being in the membrane, we have more receptors on the postsynaptic side, so the extracellular potential can be higher. But in order to make sure that it's really strengthened, biology always has safety mechanism. So you do have the insertion of new receptors, and at the same time, you have a retrograde messenger going all the way back to the presynaptic side, and that will then increase the release of glutamate. So you have both processes, you have more glutamate released and you have more postsynaptic receptors, and this ensures that this synapse is activated and stays activated. Here you see just with a different picture how it can go from LTP to LTD. So if they are stimulated at the same time, you have this left pathway here, that you have LTP long-term potentiation because there are more AMPA receptors inserted into the membrane. If the presynaptic side is very often active, but never the postsynaptic side, you have actually the opposite process. You have not a phosphorylation of signaling pathways, you have a dephosphorylation of the signaling molecules, which means AMPA receptors are removed from the membrane. There's no retrograde signaling, ensuring the higher release of glutamate into the synaptic cleft. If you, as a textbook does it, if you do it with X, if you have LTP, you have in the end more X in the membrane. If you have LTD, you lose a couple that are destructed. Actually, they are move out of the synaptic cleft, and this is part of the mechanism. So again, a very complicated mechanism, and only a part of the signaling pathways are inserted here. And as I said, my lab is still working on it. We are in particular interested in the brain-derived neurotrophic factor here, because this factor can lead to the strengthening of synapses, but it can also change the structure, because Mr. Hebb has also predicted a second thing. He said there should also be some structural changes, growth processes, he called them. And it took until 1999 until they have actually been reported. And actually, I was in the lab at the Max Planck Institute at that time in Martinsried when one of my co-doctoral students was working on it on a second day of Christmas when it was allowed to work at Max Planck, that he could observe such a thing, and a year later it was published. Here you see predicted was that when LTP occurs, the long-term potentiation, around 30 minutes later, I have the growth of new synapses. So here again, you see a pyramidal neuron. You see here the dendrites, which have these protrusions here, which are easy visible with a 2-photon laser microscope, and you can actually now observe in an experiment if you induce LTP, if there's a growth process. This is the original first recording of such an event. So it's fluorescently with GFP labeled. You see the intracellular recording here, where the red one is. There have been 30 times activation of pre and postsynaptic site, and here it's the red arrows, new synapses actually grew. So that was the first time proven that also structural changes happens, and since then it has many times been shown. Just to show you an overview, you can see here the condition before, here 30 minutes after, and here at the end of the experiment. And it has now been shown with in vivo recordings that these new synapses can be stable for weeks, months, probably even for years. That's not clear, but at least for as long as people can record, we do have structural growth processes. But not only new synapses are born, also the existing synapses become stronger. The more long-term potentiations they see, the stronger and larger they become. The more they are stimulated out of sync, the smaller they come until they evaporate. And that's also second part of what we study in my lab. What are the molecules inside the neuron that mediate these structural changes, probably initiated by the molecule BDNF, by the brain-derived neurotrophic factor. And you see here for the enlargement and for the reduction, it all boils down to the actin cytoskeleton, which is the inside cytoskeleton, the main component of, in this case, spines and dendrites. And actin binding proteins are important to mediate these changes in order to make them happen, to let a synapse grow or shrink. And here I was just writing a news and views about a paper that showed that for both processes, the brain-derived neurotrophic factor released by the postsynaptic side is important. It has presynaptic action, and it acts on the same dendrite, on the same synapse, in order to promote growth processes and also to promote changes in transcription and translation in order to make these changes, at least for some long-term memories, permanent. I didn't go into it, but that's a different thing. Let me stop with a quote from Kamoni Kachal in his recollections of his life. I noticed that every ramification, dendrite or axonic, in the course of formation passes through a chaotic period, so to speak, a period of trials during which they are sent out as random experimental conductors, most of which are destined to disappear. What mysterious forces precede the appearance of the processes, promote their growth and ramification, and finally establish those protoplasmic kisses, the intercellular articulation, which seems to constitute the final ecstasy of an epic love story. He describes the birth of a synapse. I thank you for your attention, and I think we also have a few minutes for questions and discussion. Thank you very much for having me here.
I'm sorry, everything is so interesting. I feel I have many questions. Thank you so much. That was a very interesting lecture and overview of this subject. My question is, imagine that one of us, communication engineer, wants to help you with part of your research. And let's say that the overall objective is very far-fetched, but let's imagine that we want to be able to reconstruct the information that is contained on a network of neurons only on the basis of the observation of the structure and the functionality. So following the what happens with all these processes. So first of all, do you think that will be possible? And then where should we start?
Yeah, I think it should be possible. I think what would be important is, and what's also very confusing for the field is, I think it would be very good to distinguish between the core process and modulatory processes. So which means, which is the most simple signaling pathway that you need in order to induce change and to keep change? And what then are auxiliary, what are modulatory factors? Let me tell you just one modular factor. We all know from experience, if we are motivated to learn something, it's easier to learn. Which means on the molecular level, there are molecules that actually promote change. We also have certain memories that you should actually not change. I mean, whenever I hear a new name, I should not forget my own. On the other hand, I have to incorporate the new names into my lexicon, into my list of words. And the trouble is how to keep permanent memories, but yet still allow to have new memories to happen and to have certain situations when it's easier that these processes appear and others where it's very tough and very hard because the brain is very resistant. For example, for all our routines, for all our subconscious processes, it's very hard to change them. But they all depend, that's at least what the field believes, on the same core process. So the same core process can actually be, let's see, filled into concrete. So it's extremely hard to change. Think about people um, who are addicted to certain drugs. They would love to change their behavior, but they can't. Why can't they? Because the synapses cannot change. Why is that? So I think for that, it would be very interesting to distinguish between the core process itself and then all the modulatory processes. So basically to predict, okay, I have a core process, but then what will happen if I change something else in the process? Let's say, for example, all the extracellular molecules. What happens if synapses cannot change their structure? Because the actin binding protein that would promote it is simply not expressed anymore. Because for whatever biological reason, you have a stress factor or life-threatening situation, and the network decides, well, this has to be memorized for life. Unfortunately, it's the blue color of the car that you drove into, and every time you see, or the green car, and every time you see something green, you get frozen, which is then, of course, maladaptive, but it's very adaptive to avoid a lion that you escaped once. So I think that would be an interesting property to do, and it's also very My field is one of these highly researched because it leads all the way to Alzheimer's disease, to learning and memory, to mental processes important to us humans. But it's also a field where people fight because everybody finds a molecule that's important for learning. And then you say, well, every knockout mice you produce where a molecule is missing, they have a learning deficit. That's not very helpful because people have never really defined what is a core process. So that would be interesting.
So because I think from our side, we probably can provide a kind of a holistic view of the thing where you provide like this fine detail, and then we can see everything a little bit from a distance.
Yeah, that would be helpful.
Right. Yeah. So I want to encourage collaborations.
Yeah, we started.
It's great. And yeah, so I want to give the opportunity to others to ask.
Yeah,
Thank you so much.
Sure.
Yeah, thanks again for this great talk. Very inspiring with lots of questions. So in information theory, you are used to measure the amount of information that we have in a statistical ensemble. And I know that this is also applied to your field. On the other hand, now you explained that the information is contained in a stochastic process that is somehow happening there. So do you think for which information theoretic measures you think are suitable to describe the amount of information in your neural network?
It's very tough. One thing one can ask is how many different states can a single synapse have? You can have one on and off, but actually you can have a lot more. You can have 26 different states on each single synapse. That's what Mr. Sanovsky at the Salk Institute in California calculated. I actually don't believe the number. I could have come up with 10, 20, 30. The important thing is it's not just 0 and 1, which means each single synapse has a storage capacity of a lot more than the most simple component of a computer chip. There we have 0 and 1, and here we have 26 different states. In addition to that, we can also change the structure, and that's why your question goes right into the heart, because in the moment when you change the structure of the network, how do you measure information flow? I'm sure one can think about it. You can think about more cables, more connections, more whatever. But this is changing constantly, so that would be another thing. Then, I mean, what we now have a good estimate is about what is the size of a network that you need to store a memory. For example, there's a snail which has 22,000 neurons, the Aplysia, and they can store sensitization, habituation. They do have long-term memory, so you don't need that many neurons in order to store memories. How much you need in the hippocampus is actually not clear, because unfortunately the hippocampus does not have a nice map. But people have now looked at the number of neurons that get activated when you experience something that the brain is memorizing. You can actually reactivate that later. That goes into the thousands of neurons, which means hundreds, thousands of synapses. And I think from that point, if you do these kind of calculations, I think you can do also information theory. You said it's a stochastic process. Is in a way true, but it's also one where the system makes sure it's actually very error prone. So each single vesicle has 400 neurotransmitters. You have at least 10 that are released. So in the end, if it is 8 or 12, has an effect on the signal strength. Then actually what is also, I didn't show because it became too complicated, you not only have the pre and the post-synaptic side, you also have glia cells surrounding it that kind of suck away the neurotransmitter. Even that you can change. Which means in terms of information theory, you have another complexity layer that you can actually adjust. We actually recently published a paper that when there's neuroinflammation, which is very important or detrimental and fires Alzheimer's disease, you have a process where the glutamate is not taken out of the synaptic cleft, and then you get into a situation of excitotoxicity. So not of memory, you have too much of the glutamate, and then you have too much calcium entering the cells, and the cells kill themselves. So that's another thing one could look at. But what is hard is sometimes I even use the number we need 120 bits per second in order to have consciousness. Oh, how did I come to this bit? Why not bytes? Why not 1,200? And I think this is guesswork, because what on a biological system one can, in analogy, put into a technical system, that would actually be something you guys could look into. We are just guessing. So that would be my answer, which is not really an answer, but just giving the scope of complexity. So I actually think it can be calculated, but I'm sure it will not be done by neuroscientists.
So thank you very much for talking.
Sure. Thanks for having me here.